Okay, thank you for your presentation. Yes, I'm Sofia, PhD student at the University of Udine, but I'm working on a Tropicsafe project in creative viticulture and enology in Conegliano. Our project is about looking for the genetic traits that are responsible for the resistance to grapevine yellows. Grapevine yellows, we do huge losses in grape and wine industries, but it has observed that there are differences in susceptibility among grapevine variety and also among grapevine clones of the same variety. These differences suggest the presence of specific genetic traits that are responsible for the lower susceptibility to the phytoplasma or possible to its vector. So to find out these genetic traits, we are taught and developed two different approaches. The first one is based on the differences in susceptibility among grapevine variety, the Chardonnay, the very susceptible, and the second one, Tokai Friulano, the scarcely susceptible. The second approach is based on the difference in susceptibility among grapevine clones of the same variety, the Chardonnay. So with the first approach, we want to identify the quantitative trait loci by genotyping and phenotyping one segregating population. With the second approach, we want to study the whole genome of 3 Chardonnay clones with different susceptibility and the transcriptomic profile on the early stage of the infection. About the first approach, so we cross two grapevine varieties, Chardonnay and Tokai Friulano, with different behavior to grapevine yellows, and with the F1 genotype, the segregating population, we are uncovering the quantitative trait loci. How to do that? By genotyping and phenotyping this population. Genotyping means find out the markers from the genome of the population. I use them to create a linkage map. So here I summarize the step to genotyping the population. We first extract DNA, we screen the self-fertilized plant to exclude them, and then we choose the genotyping by sequencing method to highlight the single nucleotide polymorphism that we have to use to create the linkage map. Phenotyping means evaluates all phenotype of each F1 genotypes. So we have to evaluate the susceptibility of each individual of the segregating population. To do that, we have first multiplicate and plant F1 genotypes. And then we have to infect them with infective vectors and then evaluate all the plants that we have infected by visual and laboratory analysis. Okay, here I reported the steps with more detail about the genotyping. We start with a group of F1 individuals, and with the simple sequence repeats, we exclude the self-fertilized plants. So then we use that plant, 184 F1 individuals and their parents, of course, Chardonnay Tokai, and we use that to do the genotyping by sequencing performed by Elshire group. Then we reach a big amount of single nucleotide polymorphism that we filtered to create a high quality data set with SNP that we use to create the linkage map. The linkage map that we created it's made with the parameter that I reported here in the slide, and we made that with the join map software. This is our linkage map. So you can see the 19 linkage groups, the 19 chromosomes of grapevine, and we mapped with almost 300 markers, and the graphical representation is made by our package. The name is linkage map view. On the other side, we have to evaluate the susceptibility of each F1 individuals, and to do that, we have to multiply multiply 20 plants per F1 individual, and then infected them with 20 insects per plants. In 2019 and 2020, we infected 17 different genotypes. That means 300 grapevines in the first vineyard that we planted in 2018 in Alba. And then we are monitoring the rest of all the plants that we have infected, and almost the 230 plants that become infected with symptoms in 2019 and 2020. And then we planted another vineyard in 2021 in Conegliano that consists with many of the genotypes that we have. To do this approach, we have some critical issues because we have to multiply and plant all the genotypes and monitoring that. And also to genotype 17 F1 individuals, we have to genotype, in fact, 300 plants, so we are speaking about 6,000 insects. So the work is really hard. It's really huge, so it's ongoing. So we sought and developed another approach that we hope is faster, and it's based on the differences in susceptibility among 3 grape Chardonnay clones. We suppose that the Chardonnay clones have a genome more similar to each other than the genome from different grapevine variety. So we suppose that it's easier highlight the differences, even the smallest differences between the 2 clones. So we perform a whole genome sequencing, and also we couple that with a transcriptomic analysis. We choose these 3 Chardonnay clones, C1 and C5, that are 2 Chardonnay clones susceptible, and 1 Chardonnay clone, C2, that is less susceptible. And we choose a whole genome sequencing techniques that is really accurate and is a PacBio platform, and we use a hi-fi read sequencing that's really accurate to highlight also even if the smallest differences between the genomes. And then we couple the data from PacBio hi-fi with the Illumina sequencing, and the genome assembly was performed by University of Verona. Here I reported the comparison between the genomes in the first and in the second dot plot. You can see the comparison between C1 and C2 clones in the first dot plot, and C1 and C5 comparison in the second dot plot. So here I reported a comparison between the 2 genomes clones. And you can see in the solid line that is continuous, and it's represented the similarity between the clones, the genomes clones genome. In the third dot plot, you can see the C1 genome compared with another grapevine variety, the Pinot Noir genome. And you can see that the line is not continuous. It means that there are more differences between the 2 genomes that we can observe in the first and in the second dot plots. And also, we observe that the C1 genome is more similar to C5 genome than the C2 genomes. I remember that the C1 and C5 genome are the more susceptible clones, and C2 genome is the less susceptible. So then we perform a transcriptomic profile study during the early stage of infection in vineyard. we use two clones, C1 and C2, that have, of course, different behavior against Grapevine Yellow. And we use three conditions. The first one is without insect vector to evaluate the constitutive gene expression. The second one is with multivector, because we want to evaluate the genes that are induced or repressed by only the vectors. And the third thesis was phytoplasma infective vectors to evaluate all the gene expression during the infection from Grapevine Yellows and its vector. In the table, you can see the number of differentially expressed genes, and you can see that both clones have a higher number of genes that are upregulated than the genes that are downregulated, but the number of DAGs, the total number of genes that are differentially expressed between clones, it's similar. In below of the slide, you can see that there is a Venn diagram product with the Swiss prot annotations, and you can see that the C1 clone share higher number of genes than the C2 clones. It means that the C2 clones have a more differentiated response than the C1 clone, with health vectors and with phytoplasma infected vectors. We also align the reads obtained from the RNA-seq from C1 and C2 clones in one of the clone genome, the C1 in this case, and we perform the PCR analysis. So you can see that the biological replicates represented by a multivector of the C2 clone create a cluster. So now we are studying the genes that are regulated in this cluster to uncover the specific traits that is responsible for the resistance to Grapevine Yellows in the C2 clones. About this approach, the genomic assembly and the comparison of the genome is completed, and the data processing is almost finishing. Now we are doing the biological interpretation of the data and match them with the differences in the genomic. Thank you for your attention.